I am delighted to introduce Robert A. Bradway, our keynote speaker today. Mr. Bradway is the current Chairman and Chief Executive Officer at Amgen. KGI is proud to have developed a long-standing and deep partnership with Amgen over many years. Mr. Bradway holds a bachelor's degree in biology from Amherst College and a master's degree in business administration from Harvard University. Mr. Bradway is a member of the board of directors of Norfolk Southern Corporation and serves on its audit and governance committees. He is a member of the board of trustees of the University of Southern California, that's a small school over there, and is on the advisory board of the Leonard D. Schaefer Center for Health Policy and Economics at USC. He is chairman of the CEO Roundtable on Cancer, a nonprofit organization composed of executives and founded to bring solutions to cancer treatment and prevention. Mr. Bradway joined Amgen in 2006 as Vice President of Operations and Strategy and served as Executive Vice President and Chief Financial Officer from 2007 to 2010. In 2011, he was appointed to the Amgen Board of Directors and served as the company's President and Chief Operating Officer until 2012. He became Chief Executive Officer in 2012 and Chairman in 2013. Prior to joining Amgen, Mr. Bradway was a managing director at Morgan Stanley in London, where, beginning in 2001, he was responsible for the firm's banking department and corporate finances activities in Europe. In 1985, he joined Morgan Stanley in New York as a healthcare industry investment banker and moved to London in 1990. Mr. Bradway is a great example for KGI students, and we are pleased he is here today. I would also like to welcome Bob's wife, Michelle, and his sister-in-law, Jenny Swire, and his two nieces, Alice and Addie. Welcome to all of you. And it is now my pleasure to introduce and ask you to welcome Bob Bradway, Chairman and Chief Executive Officer of Amgen.
Well, good morning. I'm delighted to be here to be part of the commencement ceremony at the Keck Graduate Institute. Thank you, Shelley, for allowing me to join everyone in recognizing the achievements of the graduating class of 2016. It's an honor to share this platform with Shelley Schuster and KGI's leadership team, with Bob Curry and the other trustees of Keck, and with KGI's distinguished faculty. As I look around this morning, and as I've had a chance to wander around the campus, I've been delighted to see the proud and smiling faces of so many families of the graduates who are here today. And graduates, your families deserve to be proud, and they should be excited too. Because as they know, timing is everything in life, and you could not be entering the world of the life sciences at a more exciting time. What physics and engineering were to the 20th century, biology and the life sciences will be for the 21st century. And if this is indeed to be the biocentury, you're entering it at dawn. Part of a generation of life scientists that will have one day the opportunity to say of your careers, we were in the right place at the right time. With its exclusive focus on life sciences, KGI has prepared you well for the start of your journey into the biocentury. I know just how well prepared you are because Amgen and KGI have enjoyed a close association through the years. In fact, some 68 of your graduates either now or have worked at Amgen through the years. And some 36 or so of your students have completed their internships with our company. And in addition, as you know, many of our staff serve as lecturers or advisors to students here at Keck. And personally, I feel a sense of connection as well to the Keck Graduate Institute because 4 of your trustees, Frank Biondi, Madhu Balachandran, Bob Volterra, and Dennis Fenton have all played an important role in my career at Amgen. And I should say a few words about one of your trustees in particular, and that's Trustee Dennis Fenton. Dennis joined Amgen, as some of you know, in 1981 as employee number 10. And speaking today as Amgen employee number 46,720, I can tell you The company likely wouldn't be where it is without him. The relationship between KGI and Amgen likely wouldn't be what it is without him. And I likely wouldn't be on this podium here with you this morning without him either. Because it was Dennis who, as an executive vice president of operations at Amgen, agreed to take me onto his team and to assimilate me into Amgen after my nearly 2 decades in the financial services industry. So if, like me, you've benefited from Dennis's mentoring or perhaps made a beer or two in the Fenton fermentation lab, please join me in a round of applause for Trustee Fenton. Degrees in hand, I know many of you are heading straight to a job in life sciences and perhaps wondering what course your career may take. But just as I could never have predicted my journey from career financier to biotech exec, I hope you recognize today that there's little point in trying to chart the full course of your career. Have your dreams? Sure. Make them big? Absolutely. But just don't expect to follow a logical, rational path towards achieving them. Let luck intervene where it will. I'm sure you've heard it said that luck is what happens when preparation meets opportunity. You leave here well prepared. Of that, I'm absolutely sure. And I'm also absolutely sure that the biocentury you're entering will deal you plenty of opportunity. And so looking out at all of you this morning, I can't help but feel that I'm looking at a group that will be especially blessed with good luck. Now, I'd like to turn to this idea that we're in the biocentury and share some thoughts with you on the opportunities that lie ahead for biotech and drug innovators. In a highly technical industry like ours, it's easy to get absorbed in the details and to lose the forest for the trees. But make no mistake, because the big picture is very clear. And it's that the biotech revolution is just getting started. Fueled by new innovation and technology and data processing capability, which of course are the fruits of the enormous advances that occurred in the 20th century in physics and engineering, we're only beginning now to just tap into new ways of understanding the stunning complexity of biology, health, and disease. And nowhere is this more true than in the field of human genetics, where recent advances are enabling unprecedented precision in our ability to examine the intricate machinery of life as defined by our DNA. And with this, we're now able to study genetic variations to help pinpoint the crucial differences between normal biology and disease. So let me provide a bit of context. The Human Genome Project was completed shortly after KGI was founded, about the time that some of you were finishing your elementary schooling. And at the time, people hoped that this achievement would quickly lead to a wave of medical breakthroughs. But this didn't happen, in part because the expense of DNA sequencing simply remained too high and it took too long. Now, sequencing a complete genome at that time still cost hundreds of millions of dollars and took years to achieve. And while having one whole reference sequence seemed an exciting milestone, it simply wasn't very useful, for example, for understanding human variation or the genetic basis of disease. But something incredibly exciting has happened since then, and you need to be aware of it. I'm referring to the billionfold, that's billion with a B, billionfold increase in the productivity of our being able to read DNA at the molecular level. Now consider what this means. Only a few years ago, it cost hundreds of millions of dollars and took years to sequence a human genome. Today, at about the cost of an iPhone, and in a matter of hours, we can read the entire genome at a molecular level for any individual. And as we do this for large groups of individuals, we can compare differences between those who develop disease and those who do not. And this enables us then to interrogate at the molecular level in a way that was not feasible even a short number of years ago, the basis for disease and the basis for health. So the productivity explosion that I'm describing places DNA and the field of human genetics squarely at the epicenter of a profound revolution in the field of biology. And I feel pretty sure that nothing will be more important to the progress that we as a society make in the biocentury than our ability to understand how this molecule at the level of the single nucleotide helps us understand the diversity of life on this planet. And the exciting thing for those of you that are entering the life sciences field at this moment is that our knowledge of the human genome is only just beginning to unfold. Because of the historical cost, most studies searching for links between genes and disease didn't attempt to read the genome in full. Instead, they skimmed the genome in search of common genetic variants. But these studies weren't as useful as we had initially hoped. In contrast with rare genetic variants, common variants tend to offer hints about the biology of a disease, but they usually don't provide actionable intelligence. Fortunately, in the last few years, new ultra-fast sequencing technologies have emerged, enabling us to generate oceans of data from in-depth genomic research, which we can sift through to look for rare variants in genetic material. And what this means is that for the first time in history, we actually have the wherewithal to map the variability and function of every gene in the human body. So the scientific community, of course, is in the early stages of doing just this. And when it's complete, it will be the greatest story ever told. Now, you might wonder, will advances like this really help us begin to unravel the mysteries of disease? You bet they will. Absolutely they will. In fact, they already are. Let me take an example from cardiovascular disease. And unfortunately, cardiovascular disease remains today the leading killer of people on our planet. But recent studies, like the ones I'm alluding to in the genetics field, have shown that rare mutations in a gene called PCSK9 can dramatically raise or lower bad cholesterol and the risk for heart disease. So for example, people who have a gain-of-function mutation in this gene have very high levels of LDL or bad cholesterol. In fact, I recently met one young man who has a gain-of-function mutation in this gene, and his cholesterol was so high that he had his first heart attack at age 25. And he's not alone, unfortunately. But in contrast, people who have a loss-of-function mutation in this gene have very low levels of LDL cholesterol and very low levels of heart disease. For example, the New York Times wrote about an aerobics instructor from Dallas who had inherited a loss-of-function mutation in this gene from both of her parents. And she had exceptionally low LDL cholesterol. And aside from her exceptionally low LDL cholesterol, she was otherwise very healthy and normal. So capitalizing on this genetic insight, high levels of PCSK9 are bad, low levels of PCSK9 are good, we've recently launched a monoclonal antibody therapy called Repatha, which capitalizes on this and significantly lowers LDL, the bad cholesterol, and with it, we hope to show the risk of cardiovascular disease. So that's one example of how human genetics offer the potential to change our approach to human disease. Let's look at another. Let's take osteoporosis, a condition which the World Health Organization considers an epidemic, a condition which leads to the unnecessary fracture of bones in women around the world who've gone through postmenopausal or who have postmenopausal osteoporosis. So the question here, do genetic insights offer us potential for the progress in this area? And again, the answer is absolutely they do. And here our story starts with a rare mutation in the gene that codes for sclerostin, which can lead to bones that are so dense that they seem virtually unbreakable. In fact, there's a recorded case of a man with this mutation who drove his motorbike into a tree at a very high speed, and he survived the accident without so much as a fracture. And that incident was a vital clue to geneticists to observe that sclerostin plays a pivotal role in regulating bone growth. And incidentally, the team that discovered this rare mutation included David Gallus, who was a co-founder of KGI. And we've taken this insight and we're developing another antibody therapy to capitalize on it to try to help prevent and treat osteoporosis and address what the World Health Organization again describes as a global epidemic. Now, thinking about the future, when we look at the aging demographics of our society, there's perhaps no more important challenge for your generation than to make meaningful new therapies in the neurosciences, addressing diseases like Alzheimer's, Parkinson's, and schizophrenia. Now, we still know very little about the biology of these diseases, and absent that knowledge, the prospect for creating disease-modifying therapies to address them remains dim. Fortunately, however, human genetics are pointing us at targets that are likely to be relevant. And these are now targets that we must pursue and that no doubt those of you graduating in your generation will pursue. And shifting rather slightly away from genetics, I think as we look back on the biocentury, we'll also see that the field of Immuno Oncology is a field that will have advanced significantly during the course of your careers. And by Immuno Oncology, I'm referring to ways to engage the body's own immune system in the fight against cancer. And today, this field, which long proved elusive, is now red hot with dozens of innovative agents promising hope in the battle against cancer. So back to my point about timing, there's never been a moment quite like this one emerging in the fields of immunology and oncology. And here's another example. For well over a century, medical practitioners have known that certain viruses could, on occasion, cause regression of some cancers, a tantalizing clue that the immune system might be involved. So researchers figured that it may even be possible to engineer a virus to kill cancer but leave healthy cells intact. And following decades of frustration, the pieces are finally falling in place. In fact, last year, Amgen had the very first so-called oncolytic virus approved for treating patients with metastatic melanoma. And this is an exciting first step, and I'm sure that we'll see much more activity like this, not just from Amgen, but from others in the industry as well over the decades to come. You know, a few years ago, talking about cures for cancer seemed far-fetched. And while it's still a tall order for sure, it doesn't seem quite so far-fetched now. And the idea that some cancers, if not cured, can be managed as chronic illness already seems attainable too. So far I've talked about drug targets. And finding a good drug target is the first critical step in developing a better medical therapy. But step two is designing a molecule that engages the target effectively. And here as well, we've seen tremendous progress as the biocentury begins to unfold. When KGI was founded, monoclonal antibodies were a new and exciting tool in the biotech arsenal. Increasingly, we're moving beyond familiar structures like antibodies to engineer more sophisticated alternatives. For example, today we have fusion proteins that are made by combining fragments of two proteins into one molecule. We have peptibodies, which link the desired attributes of a peptide with the longer duration of action of monoclonal antibodies. And we have bispecific antibodies, which link the targeting region of two antibodies. And one of our so-called bispecifics, a BITE antibody, attaches to a cancer cell on one end and to a T cell from the immune system on the other. And the proximity of the T cell to the cancer enables it to recognize the cancer cell and to destroy it, giving us another weapon, another way to harness the immuno-oncology revolution to fight cancer. And in our clinical trial program, we had patients who had run out of treatment options, patients who were told there was nothing that medical science could do for them. And I thrill with the news that some of these patients are alive today because their bispecific BITE antibodies helped them to defeat their cancer. So altogether today, our toolkit at a company like Amgen consists of about 11 distinct drug modalities, increasing the odds that we can find the right tool for any biologic target that's worth pursuing. And when this industry got started, the goal was to try to copy nature by using genetic engineering to make the same proteins found in nature. But today, what we're trying to do is improve on nature by inventing novel proteins that can fight disease in highly creative ways. And I suspect during your careers, you'll find ways to manipulate proteins and other constructs in ways that we haven't even begun to fathom today. Now, after you find a great target and design a therapeutic to engage it, you still need to figure out how to manufacture it reliably. So let me just quickly share a story with you about a revolution that's unfolding in the area of bioprocessing. Now, for decades now, companies have been operating large manufacturing plants with huge bioreactors. And these large facilities are very expensive, with construction costs of $1 billion or more, and they require very, very long lead times to bring online. They operate with a high fixed cost burden, and they're not very flexible. And frankly, we can't move new products easily in and out of them. We think we're on the verge of changing all of that. To start, we've engineered new cell lines that can generate more proteins per cell than we ever dreamed possible. And we've learned how to keep these cells alive and productive at much higher densities than ever before. And this effectively means that we can make a lot more protein in a much smaller bioreactor. In other words, we can re-engineer the whole manufacturing process in our business. And we think we're on the cusp of doing exactly that at a new facility in Singapore. Here, we've packed the production capacity of a big plant into one that's about a fifth the size of one of our traditional facilities. And we've built it at about a quarter of the cost and at about half the time of a conventional plant. And we believe in turn that we can run it at a fraction of the cost. It'll use 1/4 to 1/6 of the energy and only 1/10 as much water, and it will provide a 2- to 5-fold reduction in carbon emissions and solid waste. But the really dramatic improvement for us is flexibility. In conventional plants, we have large equipment that's welded into place, and the equipment that we have in Singapore will be modular and portable, so we can easily switch between different types of equipment to make different products. And that will help us to respond to the quickly changing demands for patients around the world and enable us to continue to supply every patient every time with the medicines they need. And those of you entering this field now will, in effect, have an opportunity to rebuild the entire production base of the biotechnology industry as new technologies like those that I've just described come online. So wherever we look now in the field of the life sciences, we see prospects for exciting innovation. But the test of success for that innovation will be whether it's able to bring real benefits to patients suffering from serious illness and to a society bearing the economic burden of that illness. And I'm optimistic that all of you, can help us meet that test. As graduates, you've already made a smart decision by investing in a degree from KGI. And you're entering a world of innovation that has the promise of helping patients in untold ways in every corner of the globe. Recognize as you emerge into the life sciences that you're entering an ecosystem which includes a role for academia, for government, for industry, and for the investment community. And your career may lead you into one or even more than one of these sectors. And I hope that none of you will take for granted the health of this ecosystem, that each of you will commit to nurturing the critical elements for sustaining innovation in science, because it's the whole of the ecosystem working together which is required to foster the kind of progress that we all would like to see in the biocentury. Now, in the decades to come, we'll have the opportunity, no doubt, to liberate people from diseases and to eliminate diseases that cut life short or crush life's potential. And those who lead this quest, and I hope and expect that you will be among them, will be doing as much real good with their lives as any of us could imagine. As we gather here, thousands of other students are attending similar graduation ceremonies elsewhere. And a great many of them will be going into business and finance, where they'll soon have an opportunity to make money or help their companies save money or make money for their clients. But your opportunity is different. You'll have the opportunity to join a field where you can help save lives and make families whole again. And as you leave here, I hope you'll keep up the energy and passion that's on display this morning and help make the great breakthroughs and discoveries that will define how far we can progress in the biocentury. And as the classic admonition goes, if not you, who? And if not now, when? And let me just close with a quote from Dr. Martin Luther King, who observed that life's most persistent and urgent question is, what are you doing for others? And the career direction you've chosen will give you every opportunity to deliver an enduring and powerful answer to that question. You're entering a career where your job, where your role really matters. My congratulations again to the KGI class of 2016. I wish you every success on the mission that lies ahead of you. Congratulations.